So we are going to pick up from where we had left off before. And of course, the I click is a bit useless today, but remember, you get double points next time. All right, so picking up from where we had left off, we briefly brushed on this idea that our bodies have prokaryotic and eukaryotic cells. And I had mentioned also briefly that we have this other organ system. I'll put it here in the corner so it's not going to bother there. Okay. So we have this other organ system that is really the microbiome. So to give you an idea of where the microbiome is and the rough amount of cells that are present in each of these sites. So I put this extra slide here showing that in your mouth you have a lot of bacteria, just in case you didn't know. But then you also have it in your lungs. So in your respiratory system, you have a lot of these microorganisms. And in breast milk, there are lots of microorganisms as well. And then if you look at your skin, you look at the amount of microorganisms that you have present on the skin. Actually, the skin is quite fascinating because it's not like your entire skin has the same type of microorganisms. If you look at the microorganisms present on your face, those are totally different than the microorganisms present in the armpits or even in your hands. If you look from at the right hand in comparison to the left hand, those are also different. So you imagine that from person to person, those microorganisms are totally diverged. Okay, and of course, you're having mucous membranes in your body. You have in the GI tract. In your entire GI tract has microorganisms, especially in your gut. Okay, and you also have in the sex organs, like vagina is packed with microorganisms. And so if you're curious about what those microorganisms are doing, you can also take a look at some projects like the human microbiome project. But again, this class is on cell biology. We're not going into microbes. Okay, so I had briefly mentioned, or I didn't even get to this slide before, showing that the presence of those microorganisms is extremely important, because if you have a healthy microbiome, those are going to work for you, and they're going to help you to have a good metabolism, and they're going to basically keep you alive. But there are some microorganisms that are not very helpful, okay? And there are direct not only correlations but also causations of some microorganisms that can destabilize your homeostasis and cause diseases. So there are some bacteria in your gut that increase the chances for you to develop type 2 diabetes, for example. And they're also related to deviations in metabolism. So metabolic syndrome, they're caused by certain bacteria. Okay? And look at the date of this publication. This is relatively recent. This is only about a year ago. Okay? So with that said, we look, some very established research that also related, and not only related, but they use the population of microbiome as causation. So this is not just a talk that this could happen. This is proved and is causation that some gut microbiome can determine if you are a lean person or if you are an obese person. Okay, some microbiome directly leads to metabolic syndrome, and they really start extracting energy from sources that you couldn't even think that those would be caloric energy sources. Okay, so based on studies that are done in the microbiome, you can have a situation in which you naturally become heavier or obese, and you're trying everything to lose weight. So I mentioned an example of a study that was done not too long ago with a woman that was in her 50s, and she had C. diff infection. And we know nowadays that exchange of healthy microbiome or introduction of healthy microbiome into the intestine of patients that have C. diff and Clostridium difficile, that's the bacteria, right? And especially the bacteria that is resistant to any form of antibiotic, the most effective way of curing the C. diff infection. Well, let me just ask the class, do you know what type of disease is a C. diff infection? Who has ever heard of C. diff in the class? I will just see the back of your hands if you stick them up. C. diff, anybody heard? Okay, some people heard. All right, have you heard of antibiotic resistant C. diff infection? So that means that no matter what kind of antibiotics you try to use, the infection will not go away, and that's quite kind of bad. All right, who heard of C. diff resistant, antibiotic resistant? Okay, so we can imagine if you try to use antibiotics, you're not going to get rid of the infection. Has anybody heard of the therapy that is 90% effective on getting rid of C. diff bacteria, antibiotic resistant infection? What kind of therapy is used nowadays? Okay, any hand up? No, okay. So nowadays there are certain hospitals that use FMT, and that's the acronym for fecal matter transplant. And that's exactly what you were thinking. You get the fecal matter, which is the fancy name for poop, okay, from a healthy person, and you transplant that into the intestines of somebody who is suffering from antibiotic resistant C. diff. Yeah, fecal matter transplant, and that is about 90% effective in curing the infection. Right now, that is some amazing research and crazy type of therapeutics. Right now, if you are thinking, how is it that they are doing that, it kind of you have this gross idea in your head now. So lately, scientists are coming up with something that is not as gross, and they're coming up with a kind of a capsule that you can just swallow, and then those microorganisms will reach your intestine and do the same thing as a fecal matter transplant. Some people are calling those crapsules. You know what I mean? Well, you get the idea. All right, so microorganisms are extremely important. And then I add this slide over here, and it's briefly telling you something about a strange behavior of mice that are climbing all over this cat, and you're thinking, what's going on here? So there is this microorganism that is a parasite. It's actually a single-celled microorganism, and is a protozoa. So this is not even a bacterium anymore. Okay, so this is a more advanced protozoa microorganism, single-celled. Okay, so this parasite here is called Toxoplasma gondii. All right, anybody heard of toxoplasmosis? Let me see arms up. Toxoplasmosis, yes. Anybody know how you can get toxoplasmosis in general? Cat, yeah, from cleaning up the cat litter. Anybody has cats in the class? I mean, not in the class, but do you have a pet, a cat? Oh, I do. We do. Cats are really cool. I really love the cats. I mean, my cat decided to invade my house and make himself comfortable on my couch, so we had to adopt him. But anyway, what's the story here? So if you see those little mice climbing all over this very patient cat, something is wrong with them. They're not supposed to go towards the predator, their own predator. So something is like really odd with these mice. So what happens is that these mice are infected with Toxoplasma gondii, okay, and this parasite is goes into these mammals' brains, and they start to change the neurotransmitters that are present in this mammal brain. Okay, so one thing that happens is that these parasites are capable of increasing the levels of dopamine, and this neurotransmitter usually will give you certain ideas that you really like to seek something that is adventurous. Okay, you like some novelty type of situation. Okay, and you know eventually tend to increase your risk-taking behavior. Okay, so if you are a adrenaline junkie and you like to go into some very cool adventures, well, there's like 33% chance that you are infected with Toxoplasma gondii. Actually, worldwide, humans are infected with Toxoplasma gondii. Around 30% of humans. Now, what does that mean? Well, to give you a better idea, we have a crowded class, right? So if you turn to your right and see the person next to you, and then you turn to your left and you see the person next to you, one of you, of the three of you, is infected by Toxoplasma gondii. That is impressive, right? Now, most of infections are asymptomatic. That means you'll never know that you have Toxoplasma in your head. And then you should be thinking, how is that possible? This is a protozoa. This is a single cell organism. How is it living inside your brain? A good question, without symptoms, right? And you're like, you start to scratch your head and you're like, oh my goodness, I thought microbiome was only related to my gut or to my skin, or and now you're telling me that I might have little organisms inside of my brain. And yeah, we do. Okay, well, and you guys are in Florida. Does anybody like to go to Busch Gardens, like 30 minutes away? From no, no, actually from us it's like an hour and about 3 hours or so in Tampa, right? Anybody likes roller coasters? Yeah, I love roller coasters. Anybody likes to skydive? No, no. Well, now you're thinking, oh my goodness, risk-taking behavior. That means I'm infected with Toxoplasma. Well, not quite. Maybe you naturally just like to do these crazy things, right? Oh, I totally like those crazy things. But then look at this. Is there a causation on the tendency of other mammals, like for example us, human beings? Is is it possible that Toxoplasma makes us love cats? Have you ever heard of some crazy cat ladies? And yeah, yeah, I love my cat. Yeah, or in that, okay, you can think also in terms of risk-taking behavior and the love of cats, like this guy here. Getting very very close to a very big predator and going on safaris and looking for these huge cats just to get close to them because I totally love them. Well, it could be that Toxoplasma is directing our behavior in a certain way. And then you look at other types of behaviors or other types um, of diseases or deviation from homeostasis that you can find in people who are suffering from mental disorders, like, for example, bipolar disorders or schizophrenia, or even obsessive compulsive disorder or aggression. Those are all related to the presence of Toxoplasma in your brain. And amazingly, there are some studies that show that autopsies done in victims of car accidents, most of them, I believe it was over 75% of those show the presence of Toxoplasma in their brains. So that means when you are driving, you might have tendencies of taking more risk than um, than you would normally take if you didn't have the parasite in your brain. Okay, so this is really cool stuff because that makes you think how much of your own thoughts and your own behaviors are being guided by microorganisms that are present in your brain or in your gut and influencing your thoughts. Okay, so I'll give you another example. Like, does anybody ever cram for an exam? Like, you kind of procrastinate sometimes, and then 2 days before the exam, you start to panic, and then you start to go through those all-nighters, and you start to gather all the information you possibly can in a very short period of time. Well, I'm sure that my students never do that. You always study with the class and keep up with the pace, and that never happens. But just in case that ever happened to you, right? Think about it. What is the type of food that you start to crave when you're studying late at night? Anybody? Do I hear like junk food, chocolate, chips? Yeah, yeah. Let me see. Arms up. Who craves chocolate chips? All right. Who craves potato chips? All right. Who craves broccoli? One person. That is extremely unusual. Well, why? Well, because stress will actually kill your good microbiome. And your good microbiome makes you crave healthy foods. But those will die under stress. So under stress, you're going to bring out and you're going to allow more replication of your bad microbiome, the kind of microbiome that makes you start to develop metabolic syndrome and eventually type 2 diabetes, right? So there is a very much causation in levels of stress and development of metabolic dysfunctions. Okay, so if you want to keep yourself healthy and you keep your mind healthy and you keep your gut healthy, uh, don't cram for your exams, basically. Okay, so you have to learn how to make your body to allow you to have the best possible health and still the best possible outcome in general. Okay, so this was a very very brief idea on how important it is to actually take a look at what is happening in these life forms and these microscopic cell forms, and then associate or understand the basic mechanisms in relation to the more elaborated life forms like we are. Okay, so with that, I came to what is supposed to be the last slide for our first class that says welcome to cell biology, right? So here we have little examples of eukaryotic cells like little mushrooms, and you have primitive life forms and bacteria and little bacteria and worms and yeah, I put mushrooms here for a reason because there is so much that we are finding out now about these life forms and how much they can alter brain function, right? But anyway, we keep it concentrated now and dive back on general features of all cells, right? So now we're going to start looking at what is it that the cells need to stay alive and to maintain life, and then all the processes that are happening in order for this overall maintenance of life to occur. All right, everybody cool in the class? Everybody still alive? Okay. So let's go one by one here. All cells use DNA. All right. So all of the living cells are going to have DNA as their inherited or as their hereditary information. Okay, so they are going to store their hereditary information in DNA. Okay, now we're going to see into more details what is DNA. So basically it's a polymer of nucleotides. And then what are the nucleotides? Well, we're going to also see in more details what they are and what their structures. But basically the nucleotides are going to form the backbone of the DNA structure. And that backbone is going to be attached to sugar molecules. And here, I'm sure that everybody has seen DNA molecules before, but we're going to go into a lot more details on what is happening here. Okay, so first thing to know is that you have a building block of DNA. So your building block of DNA is your nucleotide. Okay, it's a nucleotide. So what is this? You have a sugar that is connected to a phosphate group. So when you get a sugar with a phosphate, that gets linked, covalently linked here to a nitrogenous base, then you end up with a nucleotide. So nucleotide means nitrogenous base with a sugar and a phosphate. If you do not have a phosphate, this thing is not called a nucleotide. That will be called a nucleoside. Okay, so you need nucleotides as building blocks of your DNA, as well as RNA. But here we're first looking at the DNA. All right, so then what happens? When you put nucleotides together in a polymerization reaction, you get one long chain. And when you build the complement chain of your first template chain, you are going to end up with a double or a paired type of chain. Okay, and of course this is very vague, but we're going to see mechanisms later on as well. So what happens here? You put two of these polymers together, you end up with a double-stranded DNA molecule. Okay, so when you look at how these nucleotides are pairing with each other, we are going to see that pyrimidines get together with purines. Okay, so purine and pyrimidines will get together and in a certain pattern. So adenines are going to pair up with thymines, and they're going to form hydrogen bonds. So those are going to be two hydrogen bonds they're formed between them. And then guanines are going to pair up with cytosines, and they'll form three hydrogen bonds between them. And yes, we're going to go into a lot more detail mechanisms. And once you have these double chains together, they will naturally fold into this double helix type of format, right? So what we need to keep in mind here, you have the backbone of the double helix that is made up of the sugar molecules, and towards the inside of the double helix on more or less a 90 degrees angle, you're going to have the bases that will hydrogen bond with each other. Okay, so here's your typical DNA molecule. And here would be the first question that, of course, you cannot answer right now in the class, but is going to come up on next presentation. And yes, it's a very simple question. So, other feature of all living cells, they all have to replicate their DNA. Of course, if the DNA is carrying their genetic or inherited information, if they want to keep on reproducing and keeping alive, they will need to replicate their DNA. Okay. Now, let's see here. The way that the cells replicate the DNA is basically the same for all living cells. As we're going to see, all the DNA is replicated in a semi-conservative manner, and it's also replicated from a parent strand. Of course, you only have one thing to start replicating. And what does that mean? All right, so here you have the DNA that is present in your parent cell. And what happens during replication is that these strands separate, meaning they melt away from each other. And you're going to start replicating a daughter strand together with one of the parent strand that is also called a template strand. So when you have the cell division, in case here of mitosis, that goes from one cell to two daughter cells, each one of the DNA molecules of your daughter cells are going to have an old template strand and a new daughter strand that is complementary to your old template strand. So this is what we call semi-conservative manner. So all cells replicate their DNA in semi-conservative manner. All right, so another feature of all of living cells DNAs, those are all double-stranded. Okay, so if you look at the DNA from bacterium, you're going to see that the bacterium has double-stranded DNA, but then that bacterium has circular double-stranded DNA. So this DNA is different from your eukaryotes that have linear double-stranded DNA. Okay, now if you look at a cell, at a bacterium, you see that the bacterium, okay, here is a little picture here of the bacterium. Okay, so that bacterium has the DNA in here, so I'm going to draw a little DNA here. So that DNA, remember, is double-stranded. And okay, that's the best I can do with the mouse. My pen is not working. Okay, so that's your double-stranded DNA. And now you're gonna think, why is it that this DNA is double-stranded and is circular in the first place? What are the advantages that this bacterium has by having DNA in this form when you compare to a linear DNA? Okay, so I will need some brave souls to give some ideas, and then Belle can say that in the microphone so I can hear what you're thinking. Huh?
One region of replication?
One origin of replication, that is part of a circular DNA, yeah, yeah. But is that an advantage? Okay, let's put an origin of replication right there. Well, that is not quite an advantage because this means this DNA will only start to replicate from one side, and then you have that the lambda formation and get to the other side. But that's not quite an advantage because if your bacterium would have several origins of replication, it would do the replication a lot faster. So no, it's not an advantage. Now let's think in terms of how is it that this bacterium would defend itself from pathogens? Do you have pathogens that infect bacteria? Do you think bacteria they have to defend themselves against something?
Yes, they're saying yes.
Like what?
Retroviruses?
Viruses in general, right? Viruses. So there are certain viruses. Here, let's put this crazy virus over here. I really wish my pen would work. Let me see. Okay, so let's say you have a virus here that looks like a little spaceship. Mine look very much like a crazy spaceship because I cannot use my pen. So here you have a virus that has, let's say it has some DNA, linear DNA, that it injects into the bacterium. So this guy here is a bacteriophage, okay? And then this DNA ends up here in the bacterium. Okay, double helix to the best of my drawing abilities right now. And then this DNA could really harm the bacterium. So the bacterium has some kind of defense mechanism that it makes these enzymes that are called endonucleases, okay? And the enzyme is very good at detecting linear DNA. So the enzyme goes there and starts to eat up this DNA that was introduced by this virus. So that's one defense mechanism that the bacterium has. So if the bacterium also had linear DNA, that would be self-destructive because this enzyme would chop up the bacterium own the DNA. So that's not good. So the bacteria is really, really smart, and it makes these enzymes, they are capable of chopping up invading DNA, but not attack its own DNA. So this is only the beginning of the story. So you can imagine this bacterium has this whole elaborated defense system that can protect itself and protect the integrity of its species. Okay, so besides those exonucleases, exo is for the end of the DNA, right? Beside those exonucleases, the bacteria also has other enzymes. They're called endonucleases. So those endonucleases are going to detect sequences that are in the middle of this strand of invading DNA. Okay, so those endonucleases are going to chop from the middle, not from the ends, and they're going to kill the invading DNA. Now imagine that if this bacterium is making a protein that is an endonuclease, that sequence that this thing can identify cannot be part of the bacterial sequence in the DNA. Otherwise, of course, that would be also self-destructive. Okay, so the bacterium has to be really clever in deciding what kind of endonucleases it's going to produce. And of course, the coding for the endonucleases are it's all here inside of the DNA of the bacterium. Okay. Now, besides that, this structure of circular DNA is more stable than the structure in comparison to linear DNA. So the stability of the DNA is also a plus for the bacterium. Okay? All right, so bacteria, they all have this circular DNA. So what else is common throughout the cell types? So we know that the cells have to use the DNA as a template, and that template has to be used to make molecules of RNA. Okay, so the template of DNA will make RNA molecule. So this process of making RNA out of DNA is called transcription. So the RNA molecule is called a transcript. All right. So all the cells will make these transcripts of RNAs, and what kinds of RNAs are you going to find? You're going to find messenger RNA, transfer RNA, and ribosomal RNA. So all living cells have those three types of RNA. When you look at eukaryotes, then things even get more complicated with those RNAs. So now you're going to have other types of RNAs like pre-messenger RNA and in siRNAs, there are inhibitory RNAs and SNU RNAs and nuclear RNAs and other types of RNAs that the things get very, very complicated and fancy with RNA types. So what is this RNA business here? And why is it RNA that is this transcription thing that is so useful, right? All right, so let's see what the cells are doing here. You have the template that is the DNA, and then this template is going to make a transcript. Well, first thing we said, of course, the DNA, in order to keep the species alive, it will need to replicate and make daughter cells, right? Besides the replication, so here you have your central dogma of how you use the code in the DNA to make proteins, right? So this is going to encompass a couple of processes that are general for all the cells. First thing that's going to happen, you're going to use your DNA template to make transcripts that are going to lead to RNA production, and then you're going to use the RNA to end up doing translation and getting your protein out of this code, out of this genetic code, right? So first thing that you have is transcription, in which you get the DNA as a template that makes your RNA. So in this time, this is the time that you should actually make a note here to differentiate transcription from translation, right? So transcription is when you get the DNA template and you make RNA. Translation is when you get your RNAs and you end up with protein, right? So this is the idea of your central dogma, from DNA to RNAs to protein. Okay, so everybody in this class should be very comfortable and familiar about the distinction between transcription and translation. So now looking a bit more into this RNA business, okay? So this is another kind of polymer of nucleic acids, okay? So there are certain differences between RNA molecule and DNA molecule. First difference is that the RNA molecule uses ribose as the sugar for the backbone. So it uses ribose instead of deoxyribose. That's one of the differences. That means when you're making ribose molecules or polymers, RNA polymers, you're going to have certain differences in the nitrogenous bases that are going to be incorporated. So you're going to use uracil instead of thymine for building up those polymers of RNA. Okay, so here your building blocks for your RNA, those are going to be all ribose based, and they're going to be adenine, guanine, cytosine, and uracil. When you compare that to your DNA building blocks, you're going to have also adenine, guanine, cytosine, but then you're going to have thymine instead of your uracil. All right. One very important difference between your DNA and your RNA molecule is that the RNA is usually only one strand. So usually single-stranded. It doesn't mean that it's this linear, boring molecule. It folds into three-dimensional shapes, as you can see here. It folds in itself, and it has some areas in which it has complementary base pairing. But you don't have that cool double helix, very organized type of formation in 3D. Okay? So as an example here, you have this cool guy that I think it's really looking like a person with the arms and the legs, and he has some feet. I mean, if you just use your imagination, you can just like put a happy smiley face on this RNA over here. But for some reason, this thing is not called little man shape. It's called a clove type of shape. This thing is a tRNA, okay? It's a transfer RNA, right? So when you look at the three-dimensional structural folding of this tRNA, you see that, well, it doesn't look like a person anymore, okay? So if you think about other main differences of RNA and DNA, you'll see that this blob of molecule is a lot less stable than the double helix. Okay, so less stability and difference in their bases. Those are very much standards of differences between DNA and RNA for all living cells. All right. Other thing that is very much common among all living cells is that they're going to use proteins as catalysts to push their reactions. Okay, so all living cells, they have to maintain homeostasis, they have to take in nutrients and use the nutrients for energy, and this is where all those catalysts are going to be very useful. Okay, and after they use all the stuff for energy and for building blocks of whatever they need to make in the cell, they're going to have some metabolic waste, and then they have to get rid of that metabolic waste. And here also proteins are very useful. Okay, So in order to maintain this life form or homeostasis, you're definitely going to need those catalysts, those protein catalysts. Okay, and also remember that you're going to get the proteins based on the codes that are in the DNA of the cell. All right, so every cell has to regulate how much of those proteins it's making at a particular time, depending on the metabolic demands of the cell. So all cells are going to regulate the transcription, right? If you're looking at prokaryotes, as we're going to see in a machinery of molecular scale, you're going to see that they don't really have a lot of room to regulate transcription, but still they regulate transcription. Like they regulate that in terms of operons and how those operons are controlled, okay, based on metabolic availability of certain molecules. So prokaryotes, they have control of transcription, but it's, they really have to use their imagination to control what's going on. Because remember, in the prokaryotes, you have this situation in which you don't have a nucleus. So everything that is happening inside of the bacterial cell is happening in the cytoplasm. So here is our attempt to draw the DNA again, okay? And now you can imagine you're going to have transcription happening over there, and then right after it, you're going to have translation that is going to happen over there. And those machineries, they're going to be almost stuck to each other, very close to each other. So there is very little control of this transcription and translation type of apparatus that is happening here in the simple prokaryotic cell. Okay, but then when you look at eukaryotic cells, now you're going to end up with a lot more control of these transcription methods. Okay, because now you're going to have a nucleus that is separating the template of the DNA from the rest of the cell. And remember that when you are looking at eukaryotes, okay, so now let's draw a nucleus over here. Okay, so remember here for your eukaryotes, DNA is over there and it's kind of linear, all right? So transcription is going to happen inside of the nucleus. But then translation is going to happen in the cytoplasm, right? So here you're going to have division between your transcription process and your translation process. That will give you a lot more room to control what is happening in gene expression, right? But all living things are going to regulate transcription. So here a bit more on our central dogma that says that all cells are going to make proteins based on their messenger RNA template. Okay. All right. Now, which proteins the cells make, that has to do with what type of cells that they are. Okay. So for example here, when you look at eukaryotic cells that are present in your body, you know that if you look at a liver cell and you compare that to a cell that is present on your skin, and you look at the protein profile of the liver cell in comparison to the protein profile of the skin cell, you're going to say that, of course, the liver cell is going to make a lot more different proteins than the skin cell, right? But you look at the DNA that is inside of those nuclei, and the DNA is the same. The same DNA you have in the liver cell is present in the DNA of your skin cell. Okay? So which genes get expressed or suppressed really depend on mechanisms that are specific for each cell type. And that will have to do with epigenetic mechanisms. That means mechanisms that are not part of the genetic of the DNA itself, but they act on the DNA to either suppress or allow expression of a particular gene, depending on the cell type that you are looking at, right? So what kind of proteins that are going to be expressed? You're going to have enzymes, of course. You're going to have certain machines and switches. They're going to go either on or off. You're going to have some structural proteins that will keep the cell in a certain shape. Again, if you look at a shape of a liver cell, is totally different than the shape that is in comparison to a skin cell. All right, so each cell is going to have a certain set of proteins, right?
And não sei como posso ajudar.
Oh, geez. And you're also going to have this very cool machinery that is responsible for making proteins that is part of the ribonucleoprotein apparatus or ribosomes. Okay, so all cells are going to have ribosomes. Now, ribosomes are mainly made up of ribosomal RNA, but is not only ribosomal RNA. Okay, actually, ribosomal RNA is really, really cool because ribosomal RNA thinks that it's a protein. It kind of acts like a protein. It's actually a catalyst. Okay, so the ribosomal RNA is actually indicative that very primitive life forms didn't even have proteins to work with. Right? So maybe the very primitive life forms had DNA and had RNA, and some of the RNA behaved like proteins and did a lot of catalytic functions, right? But then we don't really have any more living cells that were following that type of thoughts of functions. But we have reminiscence of those functions in the function of ribosomal RNA. All right? Ribosomal RNAs, they are mainly ribosomes, but they get together also with proteins to make these complexes of functioning ribosome. Okay, so those complexes we call them ribonucleoproteins. Okay, so ribosomes are part of all living cells. But then when you get to more sophisticated eukaryotic cells, you're going to find other machineries as well. For example, telomerases that are going to take care of the ends of those linear DNA molecules. Okay, and also spliceosomes. There are machinery that really take care of processing the RNA before it gets out of the nucleus. Again, those, if you really think about it, those are only relevant for eukaryotes because they actually have the space inside of the nucleus to do their jobs, right? If you look at prokaryotes, they can't really do that, well, because prokaryotes have the circular DNA, so telomerase that takes care of the ends of the DNAs, they have no function in prokaryotes. There are no ends in the DNA, right? Okay, but of course, we're going to look into more details later about that too. So now is just giving you a brief idea of everything that is needed for the cells to survive. All right. So all cells are going to use energy, and they need this idea of free energy for chemical reactions to take place. So this is really telling you about the Gibbs free energy. So delta G, and this thing is not going green, delta G. So who took chemistry, basic chemistry in this class? So let's check that everybody's still awake and paying attention. Let's see, general chemistry, yes. Is that a prerequisite for this class? Yes? I hope so. So when you look at Gibbs free energy, what kind of Gibbs free energy gives you spontaneous reactions?
They're saying negative?
Yes, negative. Negative delta G gives you spontaneous reactions, right? So it's the same kind of information you had before. For a reaction to take place spontaneously, you need negative delta G, right? And we know that cells are going to need a lot of energy in order to maintain homeostasis, right? Remember that homeostasis is not equilibrium, it's a constant disequilibrium, right? So you're going to need a lot of energy to keep this in this constant stage of homeostasis. Right, so other things that are very much important for cells to be alive, they need to have boundaries, right? So they need to have a plasma membrane, and that plasma membrane is very, very important as a selective barrier, okay? And not only that, not only is important for a barrier, but is also very important to hold certain molecules that are inserted, they get inserted into this plasma membrane, okay? Besides that, the plasma membrane is very important site for communication of cells. What that means is that pieces of the cellular plasma membrane, so let's draw another cell here. So here is a cell, okay. Let's make this, all right, prokaryotic cell. So this is your bacterium, okay. So here is your tiny DNA that is circular over there. So circular DNA, not the nucleus, all right, okay. So now we look at this membrane. And this membrane can butt out little pieces or little vesicles and send those little vesicles to communicate with other bacteria. I say, oh, that is really cool. So bacteria can use the cellular membrane to communicate with each other. And actually some scientists call those speech bubbles. Have you ever heard of the speech bubbles? Let me see, hands up, who heard of speech bubbles? No. Okay, so you think bacteria are not talking to each other? Okay, how many people think the bacteria are already talking to each other before coming to this class? Okay, how many people have no idea what I'm talking about? All right, honesty. Okay, so if you look at what is happening with the membrane of bacteria, they do send out these little speech bubbles to go out and communicate with other cells that can perceive what is on this little membrane that bud out, okay? And that is important not only for bacterial communication among their own species, but it's also important for bacterial communication between different species of bacteria. And is also important for communication of the bacteria to eukaryotic cells. That means now you should start thinking, wait a minute, didn't I just mention a few minutes ago that our bodies are packed with bacteria, like microbiome? If you look at your gut, it's filled up with a lot of bacteria, and the skin and mucous membrane and the mouth and everything. And now I'm telling you that a piece of the membrane of those bacteria is able to communicate with your own eukaryotic cells. And they are so tiny that some of those little pieces of membranes, of vesicles, they pass through the blood-brain barrier. And they are able to communicate to your brain. So now you should be thinking, oh, is that why I had mentioned what is it that you are craving when you're studying, you know, during an all-nighter? And I told you the good bacteria is gone and only the bad bacteria that make you crave chocolate survive. And now you should be thinking, oh, the bacteria is sending communication from my gut to my brain that I should be eating chocolate. This is really cool. And until recently, we had no idea on how that happened. Actually, even now, we don't know exactly how that is happening. But we have a better idea based on this form of communication of outer membrane vesicles from your microbiome that get all the way up to your brain and communicate with your eukaryotic cells. All right, so this is like really, really cool stuff. So the bacteria, they can talk their own language among their own species. They can talk foreign languages with bacteria of other species, And they can also talk a very foreign language to eukaryotic species. And this is all possible because of the plasma membrane. What other things that living cells need? They need carbon and nitrogen, but not in their most common stable forms, because that's basically useless. So in order to use carbon and nitrogen, some bacteria or lower life forms will actually have to break them down and fix them, right? So we have certain phototroph bacteria that use light to break down certain forms of carbon compounds of molecules, all right, for example, carbon dioxide, and they can fix the carbon in a form that is useful for us, for example, in a building block of formaldehyde building blocks for carbohydrates, right? Some phototrophs can do that, and these are a fancy name for plants or certain type of light using bacteria. Okay, they can do that. And then you have some other forms of bacteria that can fix nitrogen. So they can take this more stable form of nitrogen and transform into ammonia, so that can be used in chemical reactions, and that can be used for us to build up nitrogenous compounds. All right, so I apologize for the snoring here in the background. That's my little dog that is taking a little nap. I hope that the microphone is not detecting her snoring in the background. I hope that's not happening in the classroom. All right. All right, so when you look at overall process of life, we really see that life can be quite simple, okay? So you have certain mechanisms that are preserved, as we mentioned, throughout the living organisms, and certain bacteria that are capable of using all of those mechanisms, they actually have very simple types of genomes, and they're very small, and they can survive in very low number of proteins getting expressed. Like, for example, here in the case of this Mycoplasma bacteria. So this type of bacteria is actually a type of bacteria that lives inside of cells, the mycoplasmas. Okay, so they only survive inside of certain other cells. In this case, this is Mycoplasma genitalium that you can imagine is part of the regular vaginal flora. Okay, so they're not really harmful. Sometimes they're harmful depending on the state of the immune system of the person. Okay, all right, so this type of bacterium only makes a few number of proteins, and they survive just fine. So with that, we get into a bit more of this idea of the diversity and comparison of different genomes. So when you look at different genomes of bacterial cells, you see that they are relatively simple, and they are really diverging from a common ancestor, because most of the coding genes that are important for maintaining certain molecular machineries, they are very much similar, no matter in which type of bacteria that you're going to look at. So looking here at this simple bacterium that has a flagellum, okay, you're going to see that inside of the DNA, you're going to have, again, your codes for your proteins, your genetic codes. And you're going to see that, again, this very simple life form has a membrane, and most bacteria have cell walls. Mycobacterium do not really have cell walls, but most bacteria have cell walls, okay? And then you look at the cytoplasm, you're going to see those little ribosomes that are floating around. They are not bound to any membrane because, again, bacteria don't have those membrane-bound organelles, right? So those guys are floating around, free ribosomes in the cytoplasm. And you're going to see that based on this very simple life form, your bacterium have certain functions, and they're expressing those certain ancestral genes, right? So when you start comparing different species of bacteria and you look at their genes sequences, you're going to see that a lot of them have genes that are very much like each other, that they have certain regions that are the same. So you call those genes related homologs of each other. Okay, so that is a proof that the bacteria really came up from common ancestor life form of bacteria, right? So when you look at how closely related they are, and you're looking at the genetic sequencing, then you really can map them into certain gene families. Okay, so very close sequencing will lead to certain gene families. Now, if you're not looking at the DNA itself, but you start looking at proteins and how closely related those proteins are, again, they can also be grouped into certain protein families. So how is that study done? How is it that you can group these things together? So let's see here, for example, if you get certain gene families of ribosomes, so you're looking at ribosomal RNA, and you line them up using some basic software, you're going to see that if you look at two types of bacteria that we have here, Methanococcus and E. coli, you're going to see that there are regions in the sequence of these two DNAs. They're very, very similar. So every time that you see here a red line connecting one sequence to another, you're going to see that the sequence is exactly the same. So you look at the similarities of these two species and you say, oh, look at that, same. Oh, this one is different. Instead of a C in the E. coli, okay, there is a T in this coccus bacteria. Okay, one difference. And then you keep on looking down the line, you see same, a little bit different. So you start comparing how similar those species are based on the DNA sequence for this particular ribosomal RNA. Now, when you compare that to the DNA that you are extracting from a human cell, you're going to see that, oh, look at that. You also have very similar sequences. Look at this, very much the same. But when you look at the sequences that are not the same, in this case you do not have a line that is connecting them, you're going to see that you have a lot more gaps in this similarity than if you compare to the two bacterial species. So when you look at the level of divergence between the species, you're going to see that the farther away in the evolution scale that you are from the primitive organisms, meaning bacteria, then the farther, the more mutations you're going to have in the diversity of that genome. Okay, but again, this is not a genetics class, but just give you an idea that conservation of functional molecules is very important and is the basis of the molecular functions that is stable and throughout the species. So how is it that you get this DNA to be so diversified? How is it that you get this thing not to be the same in comparison to the bacterial genome, right? So several things can happen during the replication of the DNA that is going to give you, cell division. So, and divergence from the common ancestor. So one thing that can happen is duplication of a certain gene. So let's say when you are replicating your genome, sometimes the machinery makes a mistake and replicates one gene more than one time, and now you end up with several genes of the same gene. So this is called gene duplication. Most of the time is accidental, but then if the daughter cell has better function and can survive better than the parent cell, then the accident is kept, and now you end up with a gene duplication. Another thing that can happen is mutation. So it can happen inside of the gene. This is called intragenic mutation, in which you can swap one base or one nucleotide for another. And another mechanism that can happen is also kind of mutation, but of huge chunks of DNA. So that's called segment mutation or segment shuffling. And again, another thing can happen, and you can exchange pieces of DNA between two different cells, and that is called horizontal gene transfer. Again, we're going to have pictures for that and examples of, visual examples of exactly what's going on here. So here we go, how you get diversification. First thing that's going to happen, you can have a little mutation. So now your new gene is a little bit different than what you had before. Okay, so this is your first mechanism here. Another thing that can happen is duplication, right? So you copy the same gene multiple times. So now you have more of the codes in your daughter cells than you have in your parent cells. And then the third type of example is really segment shuffling, in which one gene exchanges a piece of itself with another gene, and now you have diversification of both of those genes. Okay, so three mechanisms here. And your fourth mechanism here, that is your horizontal gene transfer. In this case, you have one bacterium over here that is very close to a different bacterium, and then they decide to talk to each other, and they end up swapping or donating one of the genes to the next bacterium. And now this guy here has an extra piece of DNA, meaning he has an extra chromosome, and not a chromosome, it has an extra genetic material than he had before. So this is called horizontal gene transfer. And this is basically the mechanism by which bacteria gain antibiotic resistance, right, by horizontal gene transfer. All right, so here, this would be another question that we're going to have as soon as we start next class. Okay. All right, so now we get to the characteristics of eukaryotic cells. All right, so first thing that you're going to see when you look at a microscope and you're looking at an eukaryotic cell is that you're going to see a nucleus, right? So nuclear envelope, very distinct characteristic. What that allows eukaryotic cells to do is that they can start getting bigger. They can have a bigger size in comparison to your prokaryotic cells, right? Besides that, you're going to have formations of proteins that self-assemble and are capable of creating this support mechanism inside of the cell. So that's going to be the cytoskeleton support of the cell. So remember, prokaryotic cells have cell walls, and that's what is keeping the cell in one shape, and that's what makes the cell not to explode by keeping the cytoplasm together in one space. So eukaryotic cells, they're not going to have cell wall, but they're going to have inside is support system of cytoskeleton to keep the cell in a certain shape. Another thing that is very important for your eukaryotic cell is your membrane enclosed organelles, right? Remember, prokaryotes don't have that. It doesn't mean that they don't have organelles. Remember, they have those protein enclosed organelles, protein cages and little compartments made of protein that are like same sizes of viruses, those are the protein for the bacteria, protein cages of bacteria. But now eukaryotic cells are going to have membrane enclosed organelles. All right. And again, the genome of eukaryotes get to be a lot more complicated, in which a lot of the genome is not really coding for proteins. It's going to be coding for something else, like non-coding regions or inhibitory types of RNAs or some other types of RNAs. Okay. So here is a picture of a eukaryotic cell that is really showing cytoskeleton and other structures like microtubules, and it's showing you a lot of membrane enclosed organelles, and here is showing you Golgi, and here is showing you endoplasmic reticulum that has a lot of those membrane associated ribosomes, right? And here you have, the mitochondria, of course, that's very, very important for energy production and other things related to your eukaryotic cell. And we're going to look at a lot more of those vesicles and membrane and micro things as we go along. All right, so here is a slide that's telling you how is it that eukaryotes evolved from a single or from a common ancestor cell or prokaryotic cell? This is one idea of most likely what happened during this evolution of eukaryotes. So here you have an archaea bacteria that was, happily minding its own business, but then something started to happen with the cell membrane of the archaea bacteria, that the cell membrane started to fold on itself and surround the nucleoid space of the archaea bacteria. Because remember, this space here that has a circular DNA, it is on a different phase than the rest of the cell, right? As we're going to see when you look at water molecules close to negatively charged particles, you tend to have this fourth state of water. There is more like a jello type of state, right? So most likely what happened here is that the membrane started to feel this different phase that was surrounding this circular DNA, and the membrane started to circle it and further compartmentalize that different phase that was around the DNA from the rest of the phase of the cytoplasm. So if you look here at the nuclear membrane, you will see that the nuclear membrane is really this double membrane type of thing. The inside of the double membrane is very much equivalent to the outside of the cell. So that is indicative of this folding on itself mechanism. Okay? And yes, I know I'm blowing up the time now, so I'm going to stop right here. And I